Click good morning, everyone. Welcome, sir, Padmanabhan sir, and Kaustubh sir. Good morning, both of you.
Good morning. Thank you for inviting me. Hope you can hear me.
Yes, sir. Yes, sir.
Okay.
Good morning.
Good morning, sir. Shall we wait for a few minutes, sir? 2 minutes, and we will start.
Yeah. I will have some slides to present when my turn comes.
Presentation, sir. Stop sharing. Anna. Stop. You can try, sir, now. Permission. Padmanabhan sir.
No, you can share.
So try sharing it now.
And Dr. Kamala, I have sent one slide to you.
Okay.
Your mail ID. Mail ID open. Biochemistry mail ID open. Biochemistry mail ID. Biochemistry mail ID open. Zoom.
Good morning, sir. I'm Kamla here.
Yeah, so I have sent one slide to you. You can, do whatever the mail ID you have been corresponding with me. I have one slide to project when I speak.
Sir, sorry, I have not received anything, sir. When did you send it, sir?
Which mail ID did I send you? Oh, no, let me see. This is to Sunita.
Yeah, 1 second.
Yes, sir. Just
1 second, sir. Yes, sir. I got the mail. I will just try, sir. Is it seen ಅಂತ ಕೇಳಿದ್ರೆ share the slide. Just ನೋಡಿ ಅವರು view ಮಾಡಿ ಅದನ್ನ ಡಿಸೈಡ್ ಮಾಡಿ ಬಂದು ಚೆಕ್ ಒಂದ್ಸತಿ ಅದನ್ನ ಝೂಮ್ ಮಾಡಿ ಆ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡಿ ನೋ ನಾವೇ ಶೇರ್ ಮಾಡಿ ತೋರಿಸಿ ಅಂತ ಹೇಳ್ತಾರೆ. This one. Sir, can you see now? I have shared the slide which you have sent.
Yes, yes.
Okay.
Yeah, you can project it when I speak. You can
Yes,
take
sir.
it
Yes.
off.
Sure. Recording ಆಗ್ತಾ ಇದೆ. Zoom recording ಆಗ್ತಾ ಇದೆ.
ನಾಳೆನೇ ₹4,000 ನಮ್ಮ ಟೋಟಲ್
ನಮ್ದು ಸ್ಲೈಡ್ ಶೇರ್.
Wishing you all a splendid morning. Knowledge belongs to humanity and is the torch which illuminates the world, quoted Louis Pasteur. I, Smriti Suresh,
I, Anushri,
extend our warm greetings to all the honorable dignitaries and to the enthusiastic participants gathered here.
I welcome each one of you to the one-day virtual national conference on vaccines in health and well-being, supported by the Indian Immunology Society, New Delhi, on the occasion of World Immunology Day, organized by the Department of Biochemistry, Maharani Lakshmi Amani College for Women, Autonomous.
Prayer is an action, a process of talking to God in order to achieve a higher purpose. I now request Ms. Anushri N. to render the invocation.
Shri Mahaganapatim Bhaje Shri Mahaganapatim Bhaje Hum Shri Mahaganapatim Bhaje Hum Shivatmajam Shanmukhagrajam Shri Mahaganapatim Bhaje Siddhaganasevitam Vignanashakam Siddhaganasevitam Vignanashakam Shigravara प्रसाददायकम् सदयं कपिलमुनिवरदायकं गुरुसेवा सतं हेरम्भं श्री महागणपतिं भजे कामेशनयनाह्लादकं नागलिङ्गवरपुत्रं श्रीविद्याचित्प्रभानन्दराजयो गीन्द्रवन्दितं सनिपदनिपमपगामरिसरी सनिपदनिपमपगामरिसरी तनिरिस रीम परि सनि सरि मरि सनि रि सनि सद रिस निरिस परि श्री महागणपतिं देहं शिवात्मजं षण्मुखाग्रजं श्री महागणपतिं भजे गणपतिं भजे गणपतिं भजेऽहम्.
Thank you, Anushree, for making us feel the presence of Almighty among us. Light symbolizes universal truth, knowledge, and understanding, guiding our path to the future. I request all the dignitaries to join hands in lighting the lamp.
Actually, we asked just when is the thing going to start? I guess after the lunch break, right? I saw teachers, I guess I saw Jashri ma'am, not sure. I saw Vani ma'am entering. Trina. Entering.
I request all the dear participants to keep your mics muted.
Now, I request our principal, Dr. Shashikala A, to deliver the welcome address.
A very good morning to one and all present here. A healthy immune system equals a healthy body. The International Day of Immunology is dedicated to increasing global awareness of the importance of immunology in the fight against infection, autoimmunity, and cancer. Immunology research has been a major motivation in bringing about revolutionary changes in healthcare through the development of novel immunodiagnostics and immunotherapies that have saved many lives and continue to do so. Vaccines are one of the greatest medical achievements of all time. COVID-19 shows how important active immune system is as defense and how important the vaccines are. Thanks to these vaccines, we can now start to think about a return to our normal lives again. To highlight more on these aspects, we have with us Professor Padmanabhan Govindrajan, Honorary Professor, Department of Biochemistry, Indian Institute of Science. A warm welcome to you, sir.
Thank you.
Professor Rajan, Founder and Director, Protein Design Private Limited. A hearty welcome to you, sir. Dr. Chandrasekhara S., Immunologist and Rheumatologist, Founder and Director, Changre Rheumatology and Immunology Center and Research. A hearty welcome to you too, sir. A warm welcome to all faculty and students gathered here on this special occasion. I congratulate the Department of Biochemistry for organizing an event of this kind and igniting interest in the young minds for becoming the future torch bearers of immunology across the world. Thank you. And a very best wishes for all the participants for you to attend this program, and I am sure you will have a take-home message and continue to do research in the field of immunology. Thank you. Thank you.
Thank you. To welcome our principal. I deem it as my privilege to welcome our principal, Dr. Shashikala A, to today's conference. We welcome you, ma'am.
Thank you.
I invite Dr. Sunita B, Coordinator, PG Biochemistry, to brief on the highlights of today's conference.
Thank you. Good morning to one and all here. Please, all the participants, mute your mic. Good morning to one and all present here. As we all know, today is the International Immunology Day, which has been celebrated worldwide. And this is just to create an awareness about the significance of immunology in fighting against infection, allergy, and various aspects, not only in that field, even in the transplantation and also in the immunotherapy for targeted immunotherapy for cancer and various infection and immunodiagnostics also. So the discovery of vaccine has been a major vital role which has been played in all the human life or even human life, so that we have been overcoming from the many decades, we are overcoming various infections, deadly infections, including COVID. So now we would have been aware very well about the role of vaccine in protecting us from the many decades, and it has been saved the life of many millions of humans during such pandemic situations. Apart from that, to make to make this day memorable, the Department of Biochemistry at MLAC is organizing a virtual national conference on vaccines for health and well-being. So, and also part of this, we also are conducting the quiz program with the support of Indian Immunology Society, New Delhi. So in this, we are expecting, and we are also having the participation of young inquisitive student fraternity to, so that they get an awareness and also to understand the significance of immunology in our life. As we all know, the Indian Immunology Society is a premier immunology society in India, established in 1972 with the aim of promoting the role and advancement in the field of immunology through education, research, and training. IIS is constantly achieving its vision by supporting several educational institutions to conduct such conferences and other scientific activity related to immunology. So with this, I would conclude, hoping that today's event will enlighten and ignite the scientific inquisitive student participant after listening to the talks delivered by the eminent scientists. Thank you.
Thank you, ma'am. We are sure that this conference will enlighten us on the theme Vaccines in Health and Well-being. I request Dr. Kamala Arunagiri, Head, Department of Biochemistry, to give an overview of the department.
Thank you, Anushri. I take this opportunity to introduce about our department and then the activities which is happening in our department. Right, the undergraduate program of biochemistry at MLAC, Bangalore, was started in the year 1999. Since its inception, it has remained in the forefront in continuously providing quality education in the field of biochemistry, along with instilling moral values and transforming each student into a responsible citizen. The department has competent doctoral and postdoctoral faculties who have set high standards in teaching as well as in the field of research. The postgraduate program in biochemistry was started in the year 2006. Students are offered advanced level learning and practical training
in various classical and modern areas of biochemistry with utmost emphasis on hands-on training. This includes short-term projects for students under the guidance of the faculty members with the objective of enhancing the quest for knowledge, nurturing the latent talents, and kindling the scientific temper of students. Several quality improvement events like seminars, symposia, poster presentations, hands-on workshops, quiz, etc. are regularly organized, and one such event is today's. Apart from offering UG and PG biochemistry course, the department also offers UGC-sponsored add-on course in medical lab technology, a skill-oriented course which paves way for job in various reputed diagnostic laboratories and hospitals. In addition, the department also conducts skill development programs to enhance their practical skills. The quality of teaching and breadth of research in the Department of Biochemistry has attracted students from various universities and other institutions. Thank you.
Thank you, ma'am. I request Ms. Jayashree N.S., Assistant Professor, Department of Biochemistry, to introduce our chief guest, Dr. Kaustav Nayak.
Good morning, all. I am delighted to introduce our chief guest of the day, Dr. Kaustav Nayak, Joint Secretary, Indian Immunology Society, New Delhi. Dr. Kaustav Nayak is presently working as a research scientist at ICGEB Emory Vaccine Center. He has done his master's in biotechnology and acquired his doctoral degree in immunology for the work on cellular immune responses in HIV-infected tuberculosis patients at ICMR. He later worked as Scientist B, Tuberculosis Research Center, Chennai. He was a doctoral fellow at National Institute for Research in Tuberculosis and worked on immunological markers for TB. He has into his credit a number of national and international publications and also has presented his research findings at various international platforms. He is also a recipient of a number of grants and awards. To name a few: Young Scientist Scholarship at International Congress of Immunology, travel grants to China, Ireland, Croatia, UK, Singapore, USA, and Hong Kong from various national funding agencies. We are indeed privileged to have such an eminent personality in the field of immunology as our chief guest for today. I request Dr. Kaustav Nayak to deliver the keynote address.
Oh, thank you. Sorry, thank you for the introduction. And I'll just put off the video just to have a better reception. So you can project that slide I've just sent to you. So that's I'm going to so all of you we are very delighted. The Indian Immunology Society is very delighted to have such a overwhelming response from all over the country, and it's a great privilege to be part of one of them. So we all know that we are all alive and healthy just because of the immune system.
Sorry for the inconvenience, sir. I request all the participants to please be on mute unless and until you have to interact with the speaker. Please mute yourselves. Sorry for that disturbance, sir. Over to you, sir. Sir, you are on mute. We are unable to hear you. Can you please unmute, sir?
Oh. Yeah, so did you hear me too? I was just telling my thoughts about why this Immunology Day and what we all want to get out of the Immunology Day. So I was just telling that
part because you were on mute.
Yeah, so I can just share the thoughts again. So I was just telling that we all know that we are alive and healthy just because of our immune system, and immunology research presently has revolutionized the field of medical health, especially in the era of COVID-19. So let's celebrate together the contribution of clinicians, immunologists, and immunology. We want to bring together all these heads and make immunology, learning immunology more effective and enjoyable, especially at school and undergraduate level. And we are very happy that so many colleges all over India, from Northeast, from Jammu and Kashmir, from Bangalore, from 3-4 places, from Tamil Nadu, almost 30-35 organizers have come forward to conduct this meeting of International Immunology Day. So congratulations to all of you who made this possible and who are going to carry this flag forward. It's actually great privilege, because through our motto, the Immunology Society's motto is to spread immunology education among the mass, among the resource limited settings, it should go to every nook and corner of the country, especially the college students. And that's what you people are doing. We are very happy to be a part of it and happy to be contributing both intellectually and financially. So wish you all a very happy Immunology Day, and let's set the ball rolling. We have you guys have put a very meticulous program in place, and we will enjoy that. Thank you. Yeah, Dr. Kamal, I think I'm done. I mean, I just wanted to share my thoughts. It's up to you now.
I request Dr. Pradeep S. Kaushik, Department of Biochemistry, to propose the vote of thanks.
Thank you, sir, for joining us for inauguration function. Thank you so much.
John F. Kennedy says, as we express our gratitude, we must never forget that the higher appreciation is not to just utter words, but to live by them. I deem it as my honor to propose the vote of thanks in this beautiful occasion of World Immunology Day 2022 at MLAC. On behalf of the staff and management of MLAC, we express our sincere gratitude to Dr. Kaustubh Nayak for gracing this conference on a virtual mode as the chief guest and delivering the keynote address. Thank you, sir. We thank our beloved principal, Dr. Shashikala A, for her constant guidance and support in all our activities. I express my deep gratitude to Dr. Kamala, Head, Department of Biochemistry, and Dr. Sunita P, Coordinator, PG Biochemistry, who have been the motivating and supporting energy for all the departmental activities. I thank all the faculty members of the department, the non-teaching staff, supportive and technical team, student volunteers who have put their relentless efforts in organizing this program. We thank all the participants who have joined online for this national conference. Thank you one and all, and we wish you a happy World Immunology Day. I request the audience to raise for the national anthem.
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभाशिष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you for joining us. We'll be starting the first technical session. Nivedita now.
I now request Ms. Varni to introduce G. Padmanabhan sir.
Thank you, Usha. Good morning, everyone. I am honored to introduce a speaker for the session, Professor G. Padmanabhan. Professor G. Padmanabhan is the first student of Indian Institute of Science, Bangalore, to become its director. He contributed significantly to the initiation of recombinant DNA research in the country. He initiated studies on a liver cytochrome P450 gene expression, which plays a crucial role in drug metabolism. He has been working with a malaria parasite biology for the last 3 decades. His group discovered the drug surrogate pathway against a severe malaria. He also shown that curcumin from the turmeric has a tremendous potential to treat malaria as an adjunct drug and paved the way to explain the molecular basis for the action of curcumin from a turmeric as a universal adjunct drug. Sir has guided over 50 PhDs and published over 180 papers in national and international journals. His letters in Science 1998 explaining the background being India's Pokhran nuclear test was widely appreciated. Sir has written his memoir on his expression in the doing sciences in India in the year 2008 and 18. Professor Padmanabhan was a visiting scientist and a professor at the University of Chicago for over a decade. He served international organizations such as WHO and ICGEB as an expert member. His profile was covered in Nature Medicine in the year 2003. Professor Padmanabhan has played an integral part in the growth of life science and biotechnology in the country. He continued to be active in the research after his formal retirement in the year 1998. He has mentored generations of students, junior and senior scientists, and entrepreneurs. He chairs several technical committees for the government science and technology establishment. He was elected president of the National Academy of Science India in the year 2019. He has been a chancellor of the Central University of Tamil Nadu since 2018. He was awarded the UNESCO Chair in Biotechnology in 2000. He was a co-chair chair of a COVID vaccine evaluation committee of a DBT. He has received several awards, including the Civil Award of a Padma Shri and a Padma Bhushan. Sir's profile runs pages together. With his brief introduction, I will welcome Professor G. Padmanabhan to deliver a talk on COVID-19 prospective and vaccine challenges. Over to you, sir.
Can you hear me?
Yes, sir.
Share the screen.
Yes, sir, you can share the screen, sir.
Well, the other participant is sharing itself.
Yes, sir, you can share it.
It's not my slide. Can you see the slide?
No, sir, not yet.
Not yet?
No, not yet.
I said share the screen. Can you see now?
Yes, sir. Now I can see your screen.
Okay. We thank the Maharani Lakshmi Amani College for Women for inviting me to this symposium on a National Immunology Day. It's a great pleasure and honor for me. I thank Dr. Usha contacted me, and it's indeed a great pleasure because I wrote, I had to leave at 11, so I will try to be as brief as possible. I just want to give you the COVID-19 perspective and also vaccines and challenges, because I chaired the committee of PBT for vaccine development against COVID. So I'll give you a brief about the virus itself. I'm not sure you will be able to see, but I can tell you there are only 7 corona strains which infect the human. Although there are many animal viruses, 4 of them actually cause common cold. And you would have heard about almost from the 2000 onwards, every decade you have SARS-CoV. It's probably SARS-CoV-1, which is actually referred to as SARS-CoV. Then you have the Middle East MERS-CoV. And now, 2019 onwards, we have SARS-CoV. Just to briefly tell you about the virus, essentially enters the cell, living almost every cell, although people talk about lung cells and so on, it can enter almost every cell. And through what is called as the spike protein. The spike protein, I hope you can see this diagram. And you have the receptor binding domain, and all these brown or the sugars, glycans on the spike protein. And the spike protein consists of 2 subunits, S1 subunit and S2 subunit. And there is the hinge region. And it is essentially the spike S1 subunit that would bind, and then the S2 would integrate and so on. Now, a little more on the genome of the virus. It is an RNA virus, and it has got some 30,000 bases coding for 29 proteins. 4 structural proteins, and the rest are non-structural proteins, 25 of them. And actually, you will see this brown yellow line, which is what we call an open reading frame in biochemistry. That means it actually has information for 16 proteins. So it is made as a polyprotein. And this protein is cleaved, and then each protein is released. And then, these are all used for virus processing, virus maturation, virus replication, and so on. There are 4 structural proteins: the spike protein, the envelope protein, membrane protein, the nuclear capsid. These are the 4 structural proteins. The rest are all non-structural proteins. Again, the same thing in a more elaborate fashion. What is the open reading frame about? What are all the non-structural proteins? What are the structural proteins, and so on? We'll not go into detail. Now, this is the, how does the virus bind to how does the spike protein bind to the host cell? That is through the receptor or the receptor binding domain, which binds to what is called as the ACE2 receptor, angiotensin converting enzyme 2, ACE2, that serves as the receptor for the virus. So what you see here is the protruding extended attached to a protein called ACE2. Then you will see it entering the cell. I have a larger picture. You can see the virus is coated with the spike protein. That's why it's called as corona. It's like the sun. And this binds to the spike protein, the receptor, ACE2 receptor, and the virus enters the cell. And you have the viral RNA released, and the viral RNA then gets translated. the various studies told you it can code for something like 29 proteins. And it's very interestingly, it's also capable of converting the endoplasmic reticulum. As you know, it's a reticular structure, which is converted into a double membrane. People believe it protects the virus. And therefore, the virus maturation takes place here. And then when it is released, through what we call a Golgi apparatus. There is a S1, S2 domain, as I told you. It has initially there is one cleavage with a serine protease, and in the second step, you have what is called the furin site cleavage, between the S1 and the S2 domain, and that helps the release of the virus. So this is what happens: the virus enters, it replicates, and then enters, and then again infects the fresh cell, and that is how it spreads, cell after cell after cell. Now, this is again telling you the same thing, that how the virus enters and the RNA is released and the RNA is translated and various proteins are formed, assembly takes place, and then release is taking place. Now, this SARS-2 entry, this is just a repeating of what I said. It requires a sequential cleavage of the spike glycoprotein at the S1, S2. As I told you, it consists of two subunits: cleavage sites to mediate membrane fusion, and this COP2 has a polybasic insertion. This is RR means arginine. There are at least 3 arginines at that site. That is the site I told you is cleaved by furin. This enzyme furin. These are all host enzymes which cleave. And many studies, I will not get into that. The polybasic insertion is the one that endures it with a selective advantage in lung cells and primary human airway epithelial cells. And now, this is basic about the virus, and various strains are there. It became, the most virus strain that spread was the G strain. Various levels of them, GR, GH, and also, this is all of the G, whether it is the United States or whether it is India, the G strain, various levels. Now, initially, we know deaths have been, the United States a greater death, more number of people were affected. In India also, there is a large number. I have other detailed slide, how, you had wave 1, wave 2, wave 3. This gives you the picture in something like January 2021, when we thought it is over, and again, things started. But the second wave in India, this is what you see, it came down very fast, but then there was a small peak of third wave. So the latest, for example, COVID status in 2022, we have only about 3,000, 2,000 cases. We went up to 400,000 cases, and then it has come down to 3,000 cases. And there is worry because we were reaching somehow 1,000 cases per day, and it is now becoming 3,000 cases. And there is needs a lot of caution and protection that is needed. Now, how is this, all these strains, how have they been identified? Essentially identified by sequencing. And actually, the major effort came from the United Kingdom. They had a SARS-CoV-2 lineage in the UK. They set up a consortium of various institutions, Edinburgh, Birmingham, Oxford, London, and so on. And they are the ones who first identified what we call the UK variant. Because as you know, viruses, RNA viruses in particular, keep on mutating. And you will have many, many mutants which you don't even see, because these viruses won't replicate. But the ones you actually see are the ones which actually cause a problem. And that's what the ones you see. So to do a similar thing in India, India also started the Indian SARS-CoV-2 Genomic Consortia, INSACOG. This was started in December 2022, started with all institutions together, DBT, Health Ministry, CSIR, and so on, December 2020, but actually came into being only in March 2021. So all that has happened, what has happened? United Kingdom identified the UK variant, B1.1. So I don't have time to describe all the nomenclatures that go, which is a bit complicated. Then there was a South African variant. Then there is a Brazilian variant called P1. I'll briefly, so WHO started looking at all the variants that are coming. More and more, as I told you, viruses keep on mutating. And therefore, there will be a lot of variants will come. Therefore, this became UK variant, then it became South African variant, Brazilian variant, then Indian variant. So then they said you should avoid naming the countries. Therefore, the WHO introduced a classification of alpha, beta, actually alpha UK, beta is South Africa, gamma is Brazil, delta is India. So these are all, now we use those alpha, beta, gamma, delta, and there are softwares are available. The GISAD is the International Agency for Influenza Sequencing, and that has become the place where we launch the COVID sequence launch. Literally, million sequences have been launched in the GISAD actually, all over the globe. So, actually, India probably INSACOG has done something like 200,000 plus sequences, which is actually, relatively speaking, it's a very small number. So there are ways of identifying the pangolin, next strain lineage. The origin of, as you all know, these are all bat origin, maybe pangolin was involved, and then transmitted, became a human virus, and so on and so forth. These are all software names. Next strain clade, these are all names of the software which identify this automatically, because thousands and thousands of sequences are being generated. Now, how do you classify them? Therefore, there are naming systems have been developed. Those who are interested should go back to literature and find out how these naming systems have been developed actually. So WHO said that there are two categories: variants of concern, then variants of interest. Variants of concerns are those mutations or mutants that one or more of the following changes. What? Increased transmissibility, detrimental change in epidemiology, increase in virulence, change in clinical disease presentation, or decrease in effectiveness of public health and social measures, or diagnostics are not working anymore, vaccines are not as effective, or therapeutics may not be as effective. So these are all variants of concern. Then you have variants of interest. That is, they can potentially become variants of concern. At this stage, they are variants of interest because they are found all very in different countries and perhaps carry mutations. You don't know proof, there is no proof. Perhaps carry mutations that may give rise to all those issues, as I told you earlier, not right now. Therefore, they need to be watched. So there are variants of interest. These are all epsilon, zeta, eta, theta, iota, all, all these names are there. In fact, one scientist joked there will be no more Greek words, but the way this virus is done. So the mutations I'm just giving you, we can't remember. UK variant, what are all the mutations in the, as I told you, it has got something like 30,000 bases. And what are all the amino acid changes that are taking place in the UK variant, in the South African variant, the recent variant that is the Delta variant, the D614G. Biochemists would know D stands for aspartic acid, G stands for glycine. This was the first mutation that was to be discovered. And subsequently, so many mutations have been discovered. This is the Delta variant that has been spreading. It is a variant almost spread all over in India. The more than the concern has been the Omicron. Everybody would have heard from Delta to Omicron, because Omicron, the spike protein of the Omicron variant is characterized by 30 amino acid substitutions, 3 small deletions, and 1 small insertion. Notably, 15 of the 30 amino acid substitutions are in the receptor binding domain. You see, that is why I gave you a bit of an introduction of how the virus enters the cell. It has the receptor binding domain, RBD, And that is most crucial. And that itself contains 15 of the total 30 mutations. And we are all the time talking about spike protein only. Remember, there are 4 structural proteins and 25 non-structural proteins. We have not been talking much about them. Some little work is being done now with nuclear capsid and so on. But everyone, including vaccines, which I'll come to shortly, they're also all working on the spike protein. That is the one that is bound. So these are all the key amino acid substitutions in the Omicron. And the feeling is it is increasing transmissibility. But it may not be more serious than Delta, but it is spreading much more. This is one example I'm giving in the older slide, August 21. This is what NSECOG does. NSECOG will get clinical samples through the health ministry. It's a bit bureaucratic, but still sequences will be done. Therefore, as the UK variant, how does it replace? How is the replacement? And for example, in August data you see, with this 21 data, entire country was, the virus has been replaced by the Delta strain. So now Omicron is also BA.2 becomes India's dominant COVID variant. Now Delta is being replaced by Omicron. That means Delta, additional mutations are there in Omicron or Omicron, whatever you want to call. The latest is now recombinant virus. What do you mean by recombinant virus? If your cell is infected with one strain, there can be possibility of two different viruses in 2 different cells, or in the same cell you may have 2 different viruses and they may recombine. I hope biochemistry students study recombination process, and in that sense, Hcd is a recombinant virus, which has an Omicron Hcd incorporated into a delta genome. Remember, I told you about all the alpha, beta, gamma, delta, Omicron. Now, that S gene incorporated into the delta genome is found primarily in France, and Hcd and Hc has been also identified. 2 recombinant viruses have been identified, but still they are not a major issue in India. But the point I'm trying to tell you, apart from direct mutations, mutations are caused by the polymerase, slipping. As the polymerase is copying, there will be slippages and there will be mutation. Now, we are now talking about 2 different virus strains undergoing a recombination and giving rise to a new virus. Therefore, these are all concerns for the future. BA1 and BA2, this is the recombinant virus. It just tells you what are all the various mutations, N-terminal domain, receptor binding domain, S1, S2, all these, the red ones are for BA1, together is the first row, BA2 alone, you can see the mutations that are taking place. What is worry about mutation? Mutation means they may spread more. The affinity for S protein for the spike protein for the receptor may be more, they may spread more, they may be more pathogenic, not necessarily, but these are all concerns. RT-PCR sensitivity, RT-PCR has become a local word these days. Everybody even knows RT-PCR these days. I must digress and tell you a joke. It is so popular that, in the South we make sambar, and the lady forgot to put salt one day, and the entire family could not take the taste it. They said, we have lost the taste. It must be COVID. Let us go for RT-PCR. This is just a joke which people tell. RT-PCR has become so popular these days. Everybody knows what is RT-PCR. So vaccine response. These are the concerns of mutations. Because RT-PCR is what you are amplifying a particular region and then seeing whether the product identifies the product. See, in fact, the challenge with Omicron was the S gene had a mutations, and the RT-PCR could not detect it. Therefore, they said S gene drop out. That is the Omicron. So they have to develop another set of primers to do RT-PCR to identify Omicron. So there is a concern that the RT-PCR may not work. Similarly, vaccine responses may decrease. In fact, vaccine response neutralization ability has decreased. I will come to it in a minute. So ability to spread more, ability to cause milder or more severe disease in people, ability to evade detection by specific viral diagnostic tests, decreased susceptibility to therapeutic agents such as monoclonal antibodies. Monoclonal antibodies, I'm sure students know about monoclonal antibodies. These are also used for therapy because they can neutralize. For example, if you have monoclonal antibodies to the spike protein, some of them will neutralize. How effective they are, one has to find out. There are, I think, 7 or 10 commercially available monoclonal antibodies are available. Ability to evade natural or vaccine-induced immunity. This is one of the concerns, or probably one of the main concerns, that the mutations, may be vaccinated, but still will the emerging mutation have less response to the vaccine. Quickly on the vaccines that are available globally against COVID. Carbox Vaccine, I must tell you, all the vaccines globally available are all for the spike protein. Either for the spike protein or for the receptor binding domain of the spike protein. So Carbox vaccine is a subunit vaccine, can be used for children. Only co-vaccine is different. Co-vaccine, is developed by Bharat Biotech in Hyderabad. It is for inactivated whole virus. This virus strain was first identified, isolated by a National Institute of Virology, Pune. And then, inactivated, you can put it in a vector, adeno-vector, and then inactivate the virus and then inject. That means you are injecting the whole inactivated virus. That's the only vaccine. Covishield is again for spike protein, but with an adenovector platform. I hope students should find out what are this vector platform. There are different platforms through which the spike protein gene is delivered and then transcribed, and you have the protein formed in situ. And then antibodies may be raised or the T-cell immunity will be generated. Then you have Johnson & Johnson vaccine, it is AD26. Now, AD26 is, I should tell you, these are all adenovirus variants. Because there is a concern that we are all already exposed to adenovirus. So we will all be carrying adenovirus antibodies. Therefore, the vaccine which is delivered through adenovirus may not be effective. Therefore, what AstraZeneca did, which is the Covishield Serum Institute is making, they used chimpanzee adenovector. They used the chimpanzee adenovirus and then used for delivery. That is, so AD26 is one variety. There are two interesting, in my career, I have studied more than 20 mRNAs for various proteins. But I never imagined mRNA will become a very vaccine. We always thought mRNAs are unstable. But anyway, to cut the story short, there are two companies, Moderna vaccine, Pfizer, BioNTech. These are all messenger RNA for spike protein vaccines. And these are all in vitro. You can make this mRNA. They tell me, in no time you can take and then transcribe it, polymerize. And I have a slide. I'll tell you a little more. And then the messenger RNA is given, and that is one that is as you know, it can be translated into the spike protein. Novavax vaccine is a spike protein again, but they have their own adjuvant matrix cell. Sputnik is a Russian vaccine. There are two adenoviral, AD26 and AD5. Then they have also developed a single shot Sputnik 5 vaccine, AD26 alone, like earlier one, but Johnson & Johnson. Then Indian Zydus cadila, which is a DNA vaccine. That is also first time commercially released. We have also worked on DNA vaccine for rabies and so on, but no DNA vaccine has ever been used for a human. This is the first human vaccine. I don't know how it will work. That is the global scenario. Now I am giving you the Indian situation. Inactivated virus, I told you Bharat Biotech is the Covaxin, live attenuated virus. Now mRNA vaccine is what is being developed by a company called Genome in Pune. DNA vaccine is by Zydus cadila. They also attempted to do recombinant BCG, whether there was a paper in Cell that some of these non-specific protection will be there. And then you have various platforms. I only told you about the adenovirus platform, but you can have a rabies platform, measles platform, VS vesiculostomatis virus platforms. Platform on which you express the S gene. Influenza virus platform, spike protein as the antigen. These are all globally, all are using spike protein or the receptor binding. India also has trying to develop. Some of them are already released. Zydus cadila has already released the DNA vaccine. They say it can be used for children, but still do not. They had an issue of scale up of the DNA vaccine. That is an issue. Protein vaccine is the spike protein acid, recombinant by receptor binding domain is the biological E in Hyderabad. mRNA vaccine, I told you, this is somewhat very exciting from Genome, Pune. It's a self-amplifying messenger RNA. I'll tell you one slide on the self-amplifying messenger RNA and various stages of clinical trial, nasal vaccine. There are also Bharat Biotech is also trying nasal vaccine. Now, in mRNA vaccine, what I told you about Moderna and Pfizer, but what Genova is developing is a self-amplifying messenger RNA. See, you have the spike protein mRNA. Now, how do you make? You use, as you all know, DNA-dependent RNA polymerase will be there that will transcribe it to messenger RNA. But in the self-amplifying mRNA, you also have alpha virus genes. I will not get into detail, but it forms a particle which contains both RNA polymerase and DNA polymerase. That means it will not only make DNA-dependent RNA, but the RNA that is formed will form RNA. RNA-dependent RNA synthesis. That is why it becomes self-amplifying. I hope I'm clear. That means you have both these enzymes, DNA-dependent RNA, RNA-dependent RNA, in a particle. And that is why it becomes self-amplifying. And this is something very exciting. They are in phase 3 clinical trial, and I hope they will be able to come up. And there is this suggestion that unique features of saRNA, because it can self-amplify, you can even work at a very low concentration. That is the concern, actually. And very small volumes. Normally, to manufacture these vaccines, whether you have cell culture or any of these, you need, how many? 10 kiloliters, 20 kiloliters. But somebody is talking. Please turn off your mic or something like that. Mute yourself. So this is activated, very small volume of the culture is adequate. Because you have an RNA that is self-amplifying. So that is why, this is very exciting that you can have a self-amplifying mRNA vaccine. So, but India right now, we only have 2 vaccines going around. One is Covishield, that is the Serum Institute, technology from AstraZeneca. As I told you, it's the chimpanzee adenovector, which is delivering the spike protein. Virus is, of course, finally inactivated. Covaxin is the whole viral genome, as I told you. It is inactivated, which is National Institute of Virology of Pune. These are only 2 vaccines that are available in India. This is what I told you. If you want to do protein or viral vaccine, in influenza vaccine, still egg is the source. You inject the antigen and try to isolate from thousands of eggs and so on. And if it is a culture vaccine, you need big bioreactors actually. Whereas something like a self-amplifying mRNA, they say even a small volume is adequate. Culture, 100 ml is enough, or 1 liter is. Therefore, there's a great advantage in manufacturing. Quickly, so what is the current situation of vaccination in India? I think, these numbers keep changing, but looks like there are at least 2 doses you have to take. Something like 62% of the population is covered with both the doses. And single dose, 72.5% of the population has received. And now they call a third dose as booster. 2 doses may not be adequate, especially for older people like me. You probably need a third dose. It's called a booster. In US already, booster is widely prevalent. But in India, only 1.8% of the population has received the booster dose. So the challenge before India done reasonably well so far as vaccination is concerned. How to vaccinate the entire population, including children. The population above 18 years is 75 crore. And we need 1,500 million doses, if it is 75 crore, above 18. And if you are including 18 below, 12 to 18, 5 to 12, it will come like that. So that is a big challenge in India. Our population is very large. And I should tell you, despite all the talks, discussions going on, it is still Serum Institute vaccine or AstraZeneca vaccine, the one that is covering 85% of the vaccines. Bharat Biotech's inactivated viral vaccine is only 15%. So scaling up is not an issue. Disease is an issue. Therefore, I just want to, maybe another 5 minutes I will take and tell you, vaccine, more than even vaccine, hygiene behavior becomes a priority. Primary hygiene behavior to be prioritized, linked to COVID-19, hand washing with soap, respiratory hygiene, when you cough or when you sneeze, maintaining physical distancing, cleanliness in general, self-isolation and referral. This is very, very important. If somebody has symptoms and thinks there is, nowadays, RT-PCR, people come home and collect blood and do the analysis within 24 hours. They give you the results also. But then most cases are not severe. Therefore, self-isolation becomes very important. If you are not well, self-isolation and referral, and don't conceal, because you are not only hurting yourself, you are hurting the society. So these are very important. Respiratory hygiene is very, very important. I am not very sure physical distance maintaining in India is not easy. With this crowd, this population, with workers, it's not possible. But wearing a mask, I think that is possible, and that is probably would be needed. But now I find many people do not wear masks. Anyway, but this is a virus, I must tell you, you would have seen in TV and all. So many people became virologists overnight. They all became experts. But frankly, I must tell you, we still do not know about this virus. We do not know. That is where I keep saying, in my committee, you need to have biochemistry analysis. What does the mutation mean? What does the mutation mean to the virus? Does the virus replicate more? Does the virus bind more? Does the virus, entry into the cell is more? After entry, does it replicate faster? So there are many, many fundamental questions which need to be answered. Elsewhere, they are doing such work. In India, we are not. We are still doing in epidemiology, which is important. Medically, it's very, very important, relevant. But we need to understand the virus. That's why I put this picture, we are blind men trying to still groping to find out what is this virus in the body. Because you do not know. People will say you will have a fourth wave. It will become endemic and it will become like, influenza. Every winter you will have COVID-2 infection. At this point, we do not know. This is why, just, in my opinion, it just taught us a little bit of humility, because we thought we knew everything about science. We can do this, we can do that. We have artificial intelligence, we can create this. But a tiny virus, can pose a huge challenge. And we are talking about masks rather than a vaccine that is for sure protection. But it is indeed a tragedy, I must tell you. Coronavirus, how many in India, I'm not talking about the global scenario, 4 crore people, more than 4 crore people have been infected. The 500,000 people have died, but in fact, there is a view that probably actual deaths are 3 times more. It's very difficult to get proper statistics. But official statistics itself says 5.5 lakhs. But at the same time, so I know, many of you would have known, someone close we have lost. COVID has been a great tragedy, I should tell you, but life has to go on. But only thing is, we need to remember to learn to respect nature. That's very, very important. I think that's where I will stop here, and thank you so much. Yeah.
I request the participants, if you have any queries, directly you can ask or please put it in a chat box. Unmute yourself and you can ask.
I think there are no questions, I believe.
Yeah, thank
right.
you.
Thank you so much. Thank you.
Enlightening with your talk on SARS-CoV-2 and its mutated form, and a vaccine is available in the globally. Thank you, sir. Thank you very much.
Thank you. Thanks a lot. Yeah.
Thank you, sir. This is Usha here. Thank you for accepting our request on the email and giving a talk.
Right. Thank you, Usha. Thank you.
Thank you so much.
Now we'll take a tea break for 10 minutes and we'll assemble back by 11 a.m. All of you, I request all of you to stay in the meeting till 11 a.m. Later we'll continue.
பேச வேண்டியது Mama.
Sudu will coming star. The putting tar. And but it
Hello. Hello. Hello.
Good morning, sir.
Good morning. Can you hear me?
Yes, sir. Yes.
Okay.
I request Dr. Kushalata M, Department of Biochemistry, to introduce Dr. Rajan Dege, Founder, Director, Protein Design Private Limited.
Good morning, sir. Very good morning to all. It is my great pleasure and privilege to introduce Professor Rajan Dege, a renowned scientist from IISc. Professor Rajan is currently a founder of Protein Designs Private Limited. Professor Rajan obtained his MSc from University of Bombay in the year 1974 and PhD from IISc 1982. He started his professional career at IISc as assistant professor in the year 1990 and served in various capacities as chairman, associate professor, MRDG IISc. Professor Rajan published more than 70 research papers in various reputed international journals. His research work includes expression of biologically active hormones using Pichia pastoris expression system, gonadotropin receptors, etc. He completed many funded projects from DST, such as Jesse Bose Fellowship, Immunocontraceptive for Animals, etc. Professor Rajan has many honors and awards to his credit. To name a few: Alumni Award for Excellence in Research for Science of the IAC 2016, J.C. Bose National Fellowship Award, and Fellow of IAS, and also the Fellow of Indian National Science Academy. I welcome Professor Rajan for today's talk. Over to you, sir.
Okay.
Sir, you can share your screen now.
Yeah, I'm sharing it.
Yeah.
Can you see my presentation?
Not yet, sir.
What do I do? I say copy link. What do I do?
There's an option called share screen at the bottom of your screen. It's green in color. Just click on that.
I can't hear you.
At the bottom of your screen, there's a button called share screen.
Yeah, share it. In my case, it's
Yeah, just click on that.
share. Bottom, I don't see anything. No, I'm sharing option. It says invite people, copy link and send a copy. So
No, when you click on your PPT, is the PPT open, sir? Keep the PPT
it's open.
Keep it maximized. Do not minimize it. Open Zoom and then click on share screen button.
I'm already opening. The Zoom is open.
Yeah.
Okay.
Or can you quickly share it on my email, sir? I will present it from here.
1 minute. Huh? Okay, let me Zoom. I have Zoom here. And I can't see the share is on my PowerPoint file, but share screen. Okay, I got it. Can you see my?
No, not yet. No, not yet, sir.
Oh, okay, 1 minute. I'll do one thing.
Yes, sir.
Let me see if I can, Usha, I'll send
Yes,
the
sir.
presentation to you.
Yes, sir.
Okay. I'm sorry about this. We should have tried earlier. I'm sorry, I'm not able to find my um,
Sir, if you have a problem, you can quit Zoom, mail it to me, and then again join.
that's what I'm trying to do, but I seem to be running into
Close
some
the Zoom,
computer.
just close the Zoom window. That's all it is.
1 minute, huh? Oh, you want me to close the Zoom
You
window?
can close it and mail me the PPT.
Okay.
And then join back.
Hello. Hello. Can you hear me?
Yes,
Hello.
sir, I can hear you.
Yeah, the problem I have here is that the PowerPoint file is too big that it won't come through. That's the problem. So, okay, I will now this one is open.
First, open your PPT.
It's open.
Yeah.
I'm
Keep it open and then open Zoom.
yeah.
In
And
the Zoom, on the top or at the bottom, you'll have
share
yeah.
share screen, right?
Share screen. Just click on that.
It says you cannot start screen share while the other part.
Now you can do, sir. I have stopped mine now.
Can you see my slide?
Not yet. You've clicked on your After share screen, you have to just select your PPT.
Yeah, that's what I did.
No, I can't see your slides, sir.
Share my screen, desktop 1, PowerPoint. Is it PowerPoint that I have to share?
Yes, you have to share your PowerPoint, sir.
Okay.
Your PPT.
Now hold to select.
And on the right hand side corner, you have a button called share again. You have to click on that after selecting your PPT. Shall I show you on my screen once how I do it?
Gosh.
That should be fine.
Yeah, 1 minute. I have done this, but I have forgotten how to.
No issues, sir. Take your time.
I'm sorry because it's delaying the thing. I think I have difficulty in doing that.
Okay. So just see the screen. I am showing you how we should share the screen. Okay. You're able to see my screen right now?
I can see you, but not your screen.
Okay. Now you can see my screen?
Yeah, I can see your screen.
Yeah. So look at it. On the top, I am actually clicking on new share.
Where are you showing?
There's a button where there is new share. My cursor is on new share. I'm just clicking on that.
No, I can't figure it out.
Okay. Can you see a new window now?
Yes, I can see your window.
So now if my PPT is open here, so I will select my PPT here after share screen option. You can see I've selected the PPT, which has become blue in color now.
PPT here, down.
Yes.
Okay, and then
on the right hand side corner, can you see a share button?
No, I can't see. No, I'm sorry. I think
So when you once select, probably you may not see it. I'll see it. Once you select your this one PPT, it becomes like this blue. Then on the right hand side corner, you'll find an option called share. You just have to click on that option share, and you'll see the PPT.
you are viewing the thing. Yes, 1 minute. Okay, let me try again. I think you close this.
Yeah.
Let me try.
Yeah.
Okay. Now I have chosen my PowerPoint. I've chosen my file. It's on. See, the problem I have here is on the top it says share. But once I say share, it asks me question, invite people, copy link and send a copy. I'm not sure which one I have to choose.
Come again, sir. Invite people?
Invite people, copy link and send a copy.
No. You are sharing your people. It is not sharing screen. You have an option called share screen.
Yes, now I'm sharing screen. Okay, I get all kinds of things. PowerPoint, mail system, prefer Safari, unknown. Okay, I choose a PowerPoint. Allow Zoom to share your screen. Okay. I've done that. Can you see my screen?
No, not yet. So do you have any desk on your system? or team viewer?
Pardon me?
Do you have any desk or team viewer on your system?
No, I don't have.
You don't?
Can you see my thing now?
No, not yet. Can you present it without a PPT, sir?
Well, it doesn't make sense, no?
Yeah.
Well,
Is it not getting attached to Google Drive? You can attach to Google Drive.
I'm not listening. I'll give me some 10 minutes. I will go to the lab and ask Dipti to help me with the.
Sure, sir.
Yeah, isn't it? That would be simple.
Yeah, it will be simple. Yeah.
Huh? Can you give me 15 minutes? I'm sorry about taking your time.
Yeah, sure.
Okay.
I request all the participants, we will have a break for 15 minutes. We'll continue with the talk after 15 minutes. Okay. I request all the participants to stay back in the meet.
Okay.
Yeah. And you go to your presentation.
Microsoft.
Is that the one?
No, I don't know which one. No. It's working, you know.
Feature generation. Report feature generation.
Yeah. See, this is the PowerPoint is I've chosen here.
Huh. It's called
Ammani College talk.
Okay.
Okay.
So when you go to this is open. Okay. Now you go to this one and share screen again.
Where is that?
Yeah. The Ammani College talk should show up as a preview.
Have I? But I'm sure it's on my desktop only.
Mm-hmm.
Oh. Oh.
Where is it? It's on the desktop.
Yeah, why it's not there? Where did I save it? Oh, stupid of me.
If you save it on the desktop.
Save as? No.
Save a copy.
Save a copy.
On the desktop.
On my Mac.
Recent places, you go maybe the show. Or if you go to documents and go up. Yeah, you can save it in the documents or it's in your documents.
Where? But I can't find my documents on my desktop because I'm not doing it. Where is that?
You can save it on the desktop. No? Save
Yeah, why it is not allowing me to do that?
No, it'll. We can. Is your mic off, right?
Yeah.
If we. How do you? Where do you go to save as?
Go file, no?
Oh, yeah. File.
No.
File, huh? Save a copy.
Save as.
Save a copy. Huh. On in documents or on the desktop. Yeah.
Did it do?
Yes. So now if you go to your Zoom call, how do we go to the Zoom meeting?
Yeah. Why is not doing it?
Yeah, minimize this. Yeah, the Zoom meeting. It's there. The green thing. Maximize. Now if you can share screen on the desktop.
It's not. If something is not right with this. It was much easier to go for me to go there and talk to them.
Oh, yeah.
Mute.
Did I do something in this?
Ma'am, we can see the PPT. Ma'am, Deepthi ma'am, unmute yourself.
All right, can you see my slides now?
Yes, sir, we can.
So first of all, my apologies for all this delay. Maybe it certainly tells me that I'm getting older and I need help from smarter can you see me also? Okay.
Yes,
okay, so as I said, I think I'm a little rusty about the new things, so it takes time to get things going. Anyway, can I start my talk now?
Yes, sir. Yes, sir. Please go ahead.
Okay. So the question that I have posed here is whether we can control the stray dog population by a immunocontraceptive vaccine. And it's a little kind of explosive issue whether we can really control the population of not only stray dogs that are huge menace to the societies, but there are other animal populations, wild animal populations that need to be controlled, such as wild horses, elephants, kangaroos all over the world. So there is an issue about controlling the population of these animals. And whether the question that I'm posing here is, can we develop some kind of a vaccine to control their reproduction so that we can control their, eventually, the population. And so I'm going to present some of the work that we carried out in my laboratories some years ago towards exploring the feasibility of such a vaccine, and I hope people will find interesting. Pardon me?
I'm sorry.
Okay, so when you think about animal contraceptions, there are a number of prerequisites that we need to establish before we can launch such a vaccine or strategy of contraception. First of all, it has to be a humane. That means you can't simply castrate or cull the animals to control their population. The contraceptive method has to be very effective. It has to be stress safe and non-surgical. This is an important aspect because number of pet owners also would like to get their pets sterilized, and we need a very surgical methods are very expensive and people don't like to use them on their pets. So one has to be, the method has to be safe and a non-surgical, And it has to be also affordable, because the number of animals that we need to control in state is a really large number. And the pet owners also have to find the method affordable. And it should be accepted by all societies, because there are a number of issues that come up when you try to control the population of stray animals. And there are societal issues that need to be addressed. Now, when you think about immunocontraception, that means you are trying to develop a vaccine for controlling population. And this is not it's not a new concept. It has been there for almost last 50 years, and significant efforts have been carried out in India. Both my Professor Mogul did a lot of work in trying to develop a male contraceptive vaccine, and the Delhi group led by Professor Talwar has been trying to develop a female contraceptive vaccine. And so the idea is that you immunize human individuals with appropriate antigen, and they then, as long as the titer of the antibodies against that antigen are very high, you see disruption of fertility, and some kind of a contraception is affected. So immunocontraception or immunocontraceptive vaccine is an old concept. Unfortunately, for a variety of reasons, the trials that were conducted were not continued, and I don't want to go into the details of those issues, but it was not used. It didn't go to the next level of usage. So, but then we did kind of very nice work on some of these aspects of human reproduction, and a lot of protein work that went into us formed the nucleus for the idea that we could use a contraceptive vaccine to control stray dog population. Now, when you are thinking about immunocontraception, we need to identify a target antigen. Now, this target antigen should be so important that any interference in that action of the target, the antigen, should result in disruption of the gonadal function and infertility. So this is what was important to identify. Now, the immunocontraception should be also completely reversible. And once the immunization is discontinued, the fertility should be restored. And this is one of the major aspects of human contraception, because it is usually employed by healthy individuals, not sick individuals who are sick, suffering from a disease, so they can take any toxic medicine that will cure them, but will have side effects. But in case of contraception, one cannot have such side effects. So it was essential that it should be completely reversible and it should be safe. The antigen that we are talking about must be highly immunogenic, and it should elicit antibodies that can block the action of endogenous antigens here. Now, we also need to find out a method by which we can sustain the antibody titers for a prolonged period, preferably to a single administration of immunogen. Is somebody asking me a question? So these kind of issues were involved in designing the vaccine. Now, I'm just introducing here a glycoprotein hormone family, which is, there are 4 hormones in humans, TSH, follicle stimulating hormone, luteinizing hormone, and human chorionic gonadotropin. TSH, FSH are made, and LH are made by the pituitary, whereas HCG is made by the placenta during pregnancy. Now, the importance of these hormones, FSH and LH, is that these 2 hormones are the principal regulators of mammalian reproduction. Now, in all species, FSH and LH are the principal regulators, and any interference in action of these hormones, be it through antibody strategy or any other thing that you can design antagonists of these hormones, if it is effective, should disrupt the gonadal functions, and that leads to infertility. And such so many times there are situations where an individual suffers from deficiency of hormones, and this is where the IVF programs are being used, so that the patients are administered hormone and their fertility is restored. There are reports of mutations in both hormones and their receptor, which results in disruption of the gonadal function. So the point I'm making here is that the gonadotropins are the principal regulators gonadotropin. So they are the principal antigens that we can use for immunocontraception. So this is what we have done. Now let me tell you about a little bit of actions of these hormones. Now, for example, in male reproduction, the pituitary produces a gonadotropin releasing hormone. It acts on the pituitary, releases two hormones. One is LH and the other one is FSH. LH acts on the cells called Leydig cells. These are present in the testis. These are the interstitial cells which is outside the tubules, and their function is to produce testosterone in response to LH. FSH, on the other hand, acts directly on the Sertoli cells, which are present inside the tubule, which form some kind of a protective layer for the germ cells, which eventually produce the sperm. So there is the hormonal regulation of spermatogenesis occurs through regulation by testosterone and follicle stimulating hormone, and the spermatogenesis process progresses. Now, in my laboratory, we demonstrated that we can block the functions of these two hormones by using highly specific antibodies. Now, in one case, we used highly specific antibodies and administered them to adult male rats. And what we found out that the germ cells in the tubule underwent apoptotic cell death. So what it means that if you block the function of LH or FSH by antibody or by other means also like GnRH antagonists, so they undergo apoptosis and cells die. So that is how the infertility is set in these animals because of the actions of the antibodies. So in female also, in each cycle, there is a very significant differentiation that occurs in the follicles which mature during one cycle, eventually reach what we call as the Graafian follicle, which then under the so the first steps of the maturation are controlled by FSH. LH, on the other hand, acts on the mature follicle to rupture that follicle to release the ovum, which is then, if it is fertilized, can lead to pregnancy. Now, this particular function of the rupture of the follicle is controlled by LH, and which kind of regulates, also LH regulates the remaining structure that becomes corpus luteum, and that maintains progesterone production. And if the pregnancy doesn't occur, this whole thing kind of shuts off and a menstrual bleeding occurs. So that is how the cycle is controlled. And there are a large number of data from Professor Maudgal's laboratory where it was demonstrated that specific immunoneutralization of gonadotropin results in disruption of the pregnancy, termination of pregnancy in monkeys, and that was how the infertility was setting. So what we are showing here is that the gonadotropins are the most suitable antigen for immunocontraception. So active immunization of gonadotropins can be used to develop contraceptive vaccine. Now, can we develop such a vaccine for stray animal whose population needs to be controlled? And this is the question that we are trying to answer here. Again, now when you are talking about contraceptive vaccine, apart from the prerequisites I told you, we need, first of all, we need adequate quantities of the recombinant hormones, in which we solve that problem by developing a system for producing large quantities of recombinant gonadotropins in my laboratory. Now, what we require is that once the immunogen is administered to the animals, we should be able to sustain that immune response without repeated booster. Because if you are immunizing stray animals, you can't catch them every now and then and give a booster. So we need to have a situation whereby a single dose of administration of hormone immunogen should elicit immune response that will be sustained for prolonged period of time. We cannot use conventional adjuvants. Whenever you develop vaccine, adjuvants are given to the along with the immunogen, and there are some things that are not used for hyperimmunization, but those are not cleared for human or animal usage. So we need to figure out how to use different adjuvants. And as I said, the recombinant hormone availability is also important. So in many ways, we solve several of these problems, and then we thought of exploring it to employ this principle for animal contraception. Now, but as you see, immunization of animals in the field, in the wild, requires an entirely different approach of immunization that should lead to a robust and sustained immune response without frequent boosters and without large amounts of immunogen. So we need to preserve the immunogen, we need to get sustained immune response. And this is for this, our solution was to deliver the hormonal antigens directly to the dendritic cells. And this is the novel part of our work, where we devise a method by which we can deliver the immunogen or antigen directly to the dendritic cells. Now let me talk about a little bit of dendritic cells. It was discovered by Ralph Steinman at Rockefeller University, and for which he won 2011 Nobel Prize in Physiology and Medicine. Now, what are these dendritic cells? Now, dendritic cells are the principal antigen presenting cells in the immune system. So these cells, okay, and Ralph Steinman did a lot of work to show that if one delivers the antigen directly to these dendritic cells, you get a very nice, robust immune response. And there are a number of papers that he published to this point. The dendritic cells, as I said, are the principal antigen presenting presenting cells, and they can capture the antigen from variety of different locations in the body. They can internalize the antigen, process it, and then present it to B cells and T cells to get a very sustained immune response. Now, the dendritic cells capture the antigen from circulation, but can we deliver antigens directly to the dendritic cells so that even small quantities of antigen should be able to elicit a very robust immune response? And that is what we try to do. Now, These dendritic cells have several receptors present on them. These have been identified, and although these receptors have been identified many times, the ligands for these receptors are not known. So one of the receptors that we have concentrated on in our studies was the receptor called DEC205. It is a 205 amino acid kilodalton protein. It's a very large protein, and we don't know the real ligand for this, but it belongs to the mannose receptor family. It's involved in antigen uptake, processing, and presentation. Ligands are not known, as I said, and they are also efficient in cross presentation. That means to different B cells. And so we needed to have a method by which we can target the antigens directly to the dendritic cells. Now, Steinman's group has again showed that if you have the antigen that you are interested in, if it is complex or fused to antibodies specific for the dendritic cells, receptors such as DEC205, it elicits much higher humoral and cellular response to very small quantities of antigen. And this is something that I've been stressing the importance of delivering directly to the dendritic cells. So then what do we require to evolve such an approach? So what you require is antibodies that are specific for DEC205, and then we also need a method by which you can fuse the antigens to these dendritic DEC205 specific antibodies. So first of all, excuse me. All right. What we did was to get these antibodies specific for DEC205. We expressed the N-terminal domain of this DEC205, which they call it as CRFN2 domain. And again, earlier studies have shown that CRFN2 antibodies are very useful in delivering the antigens to the dendritic cells. So what we did was to first express CRFN2 protein that's what dogs CRFN2 we express in the laboratory. And we have evolved a very interesting strategy. Now, once you have a DEC205, you have antibodies, you need to have a method by which you can fuse or complex the antigen of your interest to such antibodies. And so the way we did it was to generate antibodies against the DEC205. And then through molecular cloning, we coupled it to or fused it to a streptavidin core fragment. And then we generated biotin tag antigens. Now, in this particular case, the human chorionic gonadotropin, which is similar to LH, and follicular stimulating hormone. And so what we do is we develop a complex like this, and then administer present it to the dendritic cell and immunize the animals and use it for immunization. So for that, first of all, as I said, we express the CRFN2 domain of the DEC205, and then we, to get antibodies against the DEC205, CRFN2 domain of DEC205, we screen what we call as human SCFE libraries. Now you see a structure on the screen, you see a structure of antibodies which is the variable portion is further divided into framework region and complementarity determining region. And the SCFs are formed by fusing the C-terminus of variable portion of heavy chain to the N-terminal region of light chain, producing a single chain antibody which retains the proper binding and affinity properties of the full-length antibody. So we had this Tomlinson library with us, which we use. And then we also had another library of SCFE, which is probably turned out to be a much better library to screen, because we got very high affinity binders, and we established a method by which we could screen libraries, pull out large number of the binders or SCFE specific for DIC205 and characterize them. And this is what a very large amount of work was done to screen and characterize such SCFPs. Anyway, so once we did this cloning and isolated the dendritic cell SCFPs, we cloned them into a vector which allowed us the fusion of the variable portions of heavy chain and light chain, which are fused by serine glycine linker, to a core streptavidin fragment. And so we cloned it into that, and then we purified each of the SCFBs to further characterization. And as you see, we were able to get fairly pure SCFPs in our laboratories. Now we needed to show that these SCFPs that we have obtained will actually deliver the antigen to the dendritic cells. Now how does one prove that? So for that purpose, we developed, thanks to Professor Steinman, where he was a very generous person, we got cell lines or as first vectors in which the cells were expressing the DIC205 protein on their surface. So what we do here now is you have the cell line expressing DIC205, and then you come down, incubate these with the SCFV streptavidin, which is then complex with biotin tag HCG or FSH. And then it is followed by an HCG anti or FSH antisense, followed by anti-rabbit FITC complex. So it's the same like a large multiple complex that we form. And what we demonstrated that these SCFBs can deliver the hormonal antigens onto the cell line expressing DIC205. So this is very successful, and as you can see, the red graphs or the color graphs in each case these are 6 different SCFPs that we characterized. Particularly the interesting were the SCFP 83 and 92, because they exhibited some of the highest affinities we have seen in my research time, and they all deliver the antibodies to DIC205 expressing cell line. Can they deliver the same way to actual dendritic cells that are isolated from bone marrow? So we did a lot of work with this. We developed mouse bone marrow, rabbit bone marrow, and also we did human monocytes. We were able to isolate from by process called apheresis, and we converted those human monocytes into dendritic cells through a very complex process. And then we were able to demonstrate that our SCFPs can deliver antigen onto the dendritic cells. So we have a whole range of tools that will deliver antibodies to the dendritic cells. So after all this characterization, we needed to test whether by delivering these antibodies to the dendritic cells, can we elicit the characteristic immune response against HCG? How long it will remain? Will it be effective in neutralizing endogenous hormone? Now, we wanted to work with dogs, but there are no good facilities, dog facilities or experimental dog facilities where we could conduct this research. And it's also extraordinarily expensive to carry out on dogs. So we decided to do a surrogate model, and the surrogate model that we had was rabbits, and we showed that the CRF2 region of the dog and rabbit have nearly 81% identity. So we felt it was safe to use rabbit model to test the efficacy of our immunization. So we immunized rabbits, and we again demonstrated that rabbit bone marrow can accept the dendritic cell antibodies and deliver antigen. So then we did immunization. We again SCFP, HCG or FSH complex, and then we use poly-IC, poly-LC. This is a chemical adjuvant that has been cleared for human usage, and it's a very soluble stuff available from Sigma, and it potentiates the immune response, having an adjuvant effect. So we administer these this HCG and FSH, different amounts, and we did all kinds of experiment with that. But what is important is that we administer the immunogen is a simple intramuscular injection. When we do conventional immunization in our laboratories, we do multiple site injections with Freund's complete adjuvant, and it's quite, in many ways, it's quite a crude way of doing it. But in this particular case, all that we needed was one intramuscular injection, and the immunization started. And then we monitored the titers of the antibodies against gonadotropins for days. The maximum that we have gone is about 530 days of immunization. Many times we just gave a single injection, and then we looked at how long the antibody titers remain high. And at the end of the whole 500 days, we sacrificed the animals. We looked at the serum testosterone level, and then we looked at the testicular histology to find out whether our immunization has indeed neutralized endogenous gonadotropin and disrupted the gonadal function. And so this is what we have the solve. Now, first of all, as you see here, this is a typical titration of HCG. We're using a characteristic ELISA for HCG. And as you see here, we have started from 90 days to 405 days in this particular experiment. And the titers have remained very, very high. Now, to determine the titer, what we needed to find out, what was the antibody titer which yielded us a 1 OD of absorbance. In literature, we find that people have used something like 0.1 OD, but we were rather stringent in the calculating the titers. And here we chose 1 OD as the criterion, and we established the animal antibody titer. And as you can see here, even up to 510 days, the titer in this particular case, HCG antibody titer remain high practically for 510. In this particular experiment, we had administered a booster on the day, simple saline booster on day 240, but it really didn't matter because the titer was high and remained high for a very, very long time. So although the titer remains high, we needed to demonstrate that these actually can neutralize the hormone action. And one of the ways of demonstrating the hormone action, whether the antibodies can inhibit binding of hormone to the receptor. And that is something that we have done. And as you see here, there's something like, you know, a very high titer of receptor inhibitory antibodies are seen in our experimental model, suggesting that by giving a single immunogen or a mild booster, we can sustain the antibody titers extremely high. Now, although the antibody titers were high, what is the effect on the gonads? That was the question. Now, we did a lot of other experiments to show that different animals have similar antibody titers. And one of the criterion that we used in our animals was to look at the serum testosterone levels. Now, remember, when you immunize against Hcd, it neutralizes the endogenous rabbit LH in circulation, and as a result of which the serum testosterone levels completely get depleted. And as you see here in this particular slide panel, we saw a very high antibody titer as well as complete depletion of serum testosterone levels, suggesting that endogenous hormone is completely being neutralized. And then we looked at the spermatogenesis in these rabbits. The spermatogenesis is an extraordinarily complex differentiation event that occurs in a unique milieu present inside the seminiferous tubules. Now, the seminiferous tubules are at the periphery are the Sertoli cells, which form some kind of tight junction with each other, creating very unique milieu for the germ cells to differentiate. And this is very unique cell, and we have been studying the molecular events that occur in spermatogenesis, and we're trying to correlate to the infertility cases, which we often find that spermatogenesis is disrupted. Anyway, so it's a very complex process. The part of the differentiation process which starts with the spermatogonia, which are the primitive germ cells or stem cells, which go through a phase of gonadotropin independent phase, and then the cells enter meiotic prophase, which goes through a series of differentiation events, finally results in the meiosis, which leads to a round spermatid, and which then subsequently goes through a series of transformation to form a sperm. So this is a very complex event. Further, the spermatogenesis is complicated by the fact that the cells in typical testicle, they are associated in what is known as cellular associations. I won't go into the details of that, but it's a very complex process to study. So we then decided to look at the histology of testis. Before that, we looked at simple parameters like the normal testis in adult rabbits weigh about 2.25 grams, but in case of Hcd immunized and FSH immunized, there is about something like 30 to 40% decrease in the testicular weight. And then we had a very interesting mechanism to evaluate spermatogenesis in our laboratory. Now remember, when you take a simple biopsy or take a tissue slice of testis, digest the cells, make a single cell preparation, and then stain for the DNA content of these cells. And usually you see in testis, you have usually diploid cells, you have tetraploid cells, and haploid cells. So, and then you can quantify this whole spermatogenesis using flow cytometric analysis where we stain for DNA. And as you see in this particular panel, you can see all populations of normal spermatogenesis, where this middle peak is the diploid peak, which is something, peripheral blood lymphocyte also show the same DNA content. Then you have tetraploid cells, which is very small population in the entire testicular population. And then you have a large number of haploid cells in different stages of differentiation to go into the sperm. So these are typical flow cytometer pictures of normal testis. So as you see, they are all populations of germ cells are seen. Now, so when we looked at the germ cell population of immunized animals, both HCG and FSH immunized animal, you see the tetraploid cells have disappeared. Diploid cells also have, and most of the cells that we found in the testis were broken, damaged cells. Basically, clearly indicating to us that the spermatogenesis is disrupted in these cases. So then we looked at the typical histological pictures, and now as you see here, these are control panel, and you can see the tubules are very well populated with, I think I have a high level. This one is a typical spermatogenic tubules where you see normal cells with large number of packets of sperm cells, clearly sperms present in the tubules, and they're all distributed. And in this particular tubule that you see various stages of dog spermatogenesis, very clearly is indicating the normal histology of the testis. Now, what happens when the animals are immunized? And what, as you can see, a very large vacuoles that you see, the striking feature is there are no sperms present anywhere. This is suggesting clearly that the animals are infertile. And I have a large number of sections where you see extensive disruption of the testicular cells. Okay, so this clearly suggests that the spermatogenesis, these are FSH immunized animals. And as you see, they are also both gonadotropin antibodies result in disruption of spermatogenesis. And then we did several other characteristics of rabbit spermatogenesis, and you see a clearly effect. Now, one thing what also happens, my own studies have shown that if you neutralize the hormones in adult rats or rabbits, the germ cells undergo apoptotic cell death. So we looked at the apoptotic cell death in the sections that we had earlier characterized. And this is a typical normal, non-immunized control rabbit. And as you see, there are a lot of germ cells, but we don't see any apoptotic cells. That occasionally you see here, there, but not exactly the spermatogenesis disruption. These are again another control. Now look at the sections from the immunized rabbit, and all the cells that you see here are dead cells. 1 minute. Are apoptotic. So once the new antibody hormones are neutralized, the germ cells undergo apoptosis, and we have several such sections showing that all death there. Now, so what again? So what we have now proved that this is immunization with FSH, and also we see the more or less same effect. Apoptotic this. To summarize what I have said so far, by targeting the gonadotropins to a DEC205 receptor, we were able to elicit a sustained a bioneutralizing antibodies response for a prolonged period of time. And this results in complete disruption of the gonadal function. We haven't done the fertility testing with these rabbits because we didn't have the proper controls in rabbits. We have extensive data on monkeys and probably rats, but we were not interested in working out with rats or monkeys. We couldn't really do it right now. So the idea that we have used here is that if you neutralize both gonadotropins, one can prepare an immunogen where we prepare both biotin tag, HCG and FSH, tag them to our SCFVs, and can elicit a very nice immune response that will disrupt the gonadotropin function, and it will cause infertility in animals. Now, how useful are these studies that we did it with rabbit for other animals? Now, what we have been studying gonadotropins for several years now, and usually find that there is a huge degree of homology between human down to rodent gonadotropins. And invariably, the antibodies that we generate against, now in this particular study, we use recombinant human chorionic gonadotropin and human FSH. But one can do studies with sheep LH or sheep FSH or LH or FSH from any other species. And what our experience over the years also tells us that antibodies raised against, say, ovine or human FSH often neutralize the gonadotropins from other species such as rabbits and rats and rodents and monkeys. So we are fairly certain that antibodies against human gonadotropins that we have used here will elicit antibody response that will lead to a disruption of canine gonadal function. Now, we have carried out studies only with male rabbits, but as I showed you the evidence that gonadotropins are also the regulators of female reproduction. So we are fairly certain that immunization with HCG and FSH will bring about infertility in the females also. Now, the idea of directing gonadotropins to dendritic cell will have several advantages. Obviously, we expect a very high, prolonged immune response that will be. So one single dose of immunogen administered, maybe we'll be able to sustain the immune response at least for a year and a half to 2, as we have demonstrated. So we don't need to give repeated booster to the stray animals. That's going to be one major problem that we'll have to see. How will we give a booster to an animal that has been previously immunized? And we will do a lot of studies to do that. I mean, there is a logistical issues there, but that we'll have to kind of sort it. The second thing is the amount of hormone that we use in rabbit immunization was somewhere 100 micrograms. Now, our previous experience where we used alum as the adjuvant and used in monkeys, we had to give at least a gram of hormone to the individual animal to get the characteristic response. So almost 10-fold less is required for rabbit immunization, and we are fairly certain that dog immunization will also require the same amount of immunogen. And we can think that we have a way of getting a sustained immune response to the gonadotropins, and that can be used to immunize dogs. Now, world over, there are situations where animal population needs to be controlled, particularly wild horses, where they have a population that needs to be controlled, kangaroos in Australia, elephants in Africa, you know. And the interesting feature is that all these animals require LH and FSH to sustain their reproduction. So if you neutralize, interfere in the actions of hormones using this method that we have developed, fortunately, the dendritic cell SCFs that we generated seem to be binding to multiple species. That is what we have shown. We have shown human, rabbit, dog, and so on and so forth. So we expect them to bind to the elephant dendritic cells or kangaroo dendritic cells, so that we are hoping that. So one can use the same strategy to control the populations of large number of the different animal populations. So this is where we are. We would like to carry out dog studies, but at least the dog studies require a different mechanism, and the expenditure involved is so high that we have not been able to generate the funding to carry out this. And this particular study, dog studies, will have to be also carried out in extremely controlled environment because the strategy will not only be used for wild animals, but also can be used for pet animals, where the pet owners don't have to castrate their pets. And so this can be used for dogs, cats, and any other pet that you may have for disrupting its reproductive function. What's happening? It's froze. Anyway, these remaining slides were only summary slides. So what I hope I have convinced you that we have a novel method of immunization which can be used to control the reproduction of animals, and thus we can control the population of stray dogs. I'd like to thank, I have a slide which, this seems to be frozen here. I have a slide which I'd like to thank my students and colleagues who have contributed over a long time to develop this concept and develop the reagents that we have generated. So with that, I'd like to thank you for your kind attention, and my apologies for the delay in presentation caused by my ignorance of modern computers. So again, sorry for that, and thank you for your attention. Thank you.
Thank you so much, sir, for your enlightening session. The session is now open for questions. Participants can unmute yourself and ask questions.
By the way, same strategy that we have isolated these SCFs can be also used for other antigens. I mean, we can use for infectious agents, can use them for cancer immunization and cancer antibodies. So we have generated a versatile tool to deliver immunogen directly to the dendritic cells, and that is what we think it is a very important observation.
Since there are no questions, I would like to thank sir for this wonderful session. I would request all the participants to return back to the session by 1:30. It's lunch break now. Thank you. Thank you once again, sir, for your time.
Thank you very much.
I have just logged in. If somebody can help me to check it. Yeah. Yeah, sure. Hello. Hello.
Yes, sir.
Yeah, am I audible? Yeah.
Yes, sir. You're audible, sir. We'll
Yeah.
just start in another 5 minutes.
Shall I just try once the?
Yes, sir. You can share the PPT and see.
It's visible.
Yes, sir. It is visible.
Oh, okay, fine.
So you can put it on slideshow mode and see once.
Yeah, sure.
Yes, sir. It is on slideshow mode.
All right. Okay, fine. Done.
Oh, okay.
Hello. This is ಹೇಳಿ ಮೇಡಮ್ 2:30 ನನ್ನ ಲೆಕ್ಚರ್ ಮುಗಿದ ಕೂಡಲೇ ನೋಟ್ ಕಳಿಸ್ತೀನಿ ಅಂತೆಲ್ಲ ಅದೇ ಯಾಕಂದ್ರೆ ನನ್ನ ಲೆಕ್ಚರ್ಸ್ ಟೈಮ್ ಇರುತ್ತೆ, ಹೇಳಿ ಅವ್ರಿಗೆ ಸರ್ ಊಟನೂ ಮಾಡಿಲ್ಲ ಮುಗಿಸ್
ಮತ್ತೆ ಅಳ್ತೈತೆ ಅನ್ನು. ಇವಾಗ ನೆಕ್ಸ್ಟ್ ನಾಡಿದ್ದು ನಾಡಿದ್ದು ಆಳ ನೋಡಿ ಹೆಂಗ್ ಮಾತಾಡ್ತಾನೆ. ಹಂಗೇನಿಲ್ಲ ಆದ್ರೂ ಹೋಗ್ಬೇಕು. ಯಾರ್ಗೆ ಕೇಸ್ಗೆ ಹೋಗೋಣ ಬಾ, ಈಗಲೇ ಎದ್ದು ನಿಂತ್ಕೊಂತೀನಿ.
Yeah, shall we start actually?
Welcome back to everybody. We'll be continuing with our sessions now. I request Ms. Usha T, Associate Professor, Department of Biochemistry, to introduce our next resource person, Dr. Chandrasekhar S.
Good afternoon, everyone. I am very honored to introduce Dr. Chandrasekhar sir. He has completed his MBBS in 1988 from Karnataka Medical College, followed by MD in 1992 from Mysore Medical College. He obtained his DM in Clinical Immunology from Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. He started his professional career as professor and HOD in MS Ramaya Medical College. Later, he founded Chandray Rheumatology and Immunology Center and Research in Bangalore. From past 18 years, he is the medical director and consultant rheumatologist in Chandray. He is the recipient of many awards, to name a few: IMA Pune Annual Neo Oration Award, C.V. Raman State Award for Young Scientists, Dr. Malaka Reddy Oration Award. Apart from this, he has various affiliations, to name a few. He is the president of Indian Rheumatology Association, General Secretary of the Society of Inflammation Research, Managing Trustee of the Immunology and Arthritis Research and Education Trust. He is also the executive editor of Chandray Journals and faculty and program coordinator in Immunology and Rheumatology in CRI-CR. He is the board member in ICMR, Roche Pharmaceutical, Pfizer, Johnson & Johnson Limited, Eli Lilly, and Clinigen. He has authored more than 250 articles in journals, conference proceedings, newsletter bulletins, and he's also authored more than 25 books. I now request Chandrasekhar sir to deliver his talk.
Thank you, Dr. Usha and Dr. Sushil and the entire Maharani Lakshmi Ammani's team for inviting me to share my perspective with reference to some of the immunological aspect where we are working. And of course, everyone has to work through for a better way of handling the varieties of diseases. So let me share my slides. I think my slide is visible.
Yes, sir, it's visible.
I think in next 30 to 45 minutes, I will take you through why we should start learning and working to understand the immune response to several diseases. But of course, here I will take the immune infection as one of the key disease, because either till the COVID came or even earlier to that also, we were contemplating and we were thinking that looking at the way a host respond to the infection matters when it comes to the outcome. So we really knew that if the patient rolls into sepsis, if patient develops varieties of complication to infection, the chance of him coming out of infections is less likely. We were also thinking that it is the immune system, if it doesn't resist it properly, maybe the reason for we losing the patients. We thought it's his patient's failure to respond to the infection. But we also started realizing it is not just the failed defense, it could be overreaction. That what we learned in and heard in COVID infection, that cytokine storm, or patient rolling into multiple thrombotic state, or an altered high immune response, which also could be the factor responsible for we losing the patient or patient moving to ICU and subsequently losing the patient. So that made us to realize that immune response plays a key role in how the human body overall respond or overall comes out of the disease to which he has been afflicted. So with these primary things, my objective is to figure out, make you sensitize how there is a difference in individual response to infections and what makes an individual immune competent compared to another individual's what can go wrong while fighting an infections, and role of antimicrobial vaccines, supportive therapies in fighting infections. Keeping this as the basic background, just I will extrapolate in last few minutes, how does this learning and understanding will help us in managing autoimmune disease and others. So all of us know the predominant invaders are the 5 main types. It could be virus, bacteria, fungi, protozoa, or helminths. And we also know now that the way the human body responds to virus, bacteria, fungi, protozoa, and helminths are different. So one of the factors which determines how a body would fight with the infection, it depends on the invader. So if it is virus, it's different. If it is bacteria, the way it works are different. The way the fungus can infect are different. And helminths and protozoas can affect the human tissue are different. So based on these broader perspective of invaders, the immune response is going to be different. So I don't think I will touch on that, because here the way the body responds is determined by the invader. So when it comes to the immune system, I think how we work on, I just use some of the, since the audience are little new to the immune response, I thought I'll just make them a little more sensitive to some of the terms I use. When I speak pathogens, we talk about the agents that causes the disease. And when we talk of immune system, it's a system which recognizes the foreign body and responds with the production of immune cells and proteins. But we now have realized that immune system is not just made to fight the foreign bodies and respond to the invader. It has a lot of other responsibility in bringing in the homeostasis. It tries to bring in a good homeostasis. And as a consequence of bringing in the homeostasis, it plays a vital role in responding to varieties of non-infection diseases, which even include myocardial infarction, which may also include the varieties of other minor diseases. And of course, in autoimmune disease, it's the immune system which has gone terribly wrong. But even in cancer, oncology, and in all of these things, we do find out that the immune system plays a vital and a critical role, resulting in having a major role in the way the body responds to a given disease. And we know there is something what we call as innate immunity, which is both there in the even the primitive living organisms. And of course, this innate immunity is carried up towards even the vertebrates and the higher evolved organisms. And we have something called adaptive immunity, which is unique to vertebrates, where the cells can recognize the proteins or the specific pathogens based on the previous memory or prior informations. So with that, things in way simple. So when a pathogen, such as a bacteria or fungi invades, so we have innate immune response. How does it work? You have a barrier. Its skin sees to that it doesn't allow the pathogen to enter. Mucous membrane and its secretions tries to fight out the bacteria and viruses. If it overrides it, then you have the next set of innate response, which has phagocytic cells, you have a natural killer cells, varieties of antimicrobial proteins and inflammatory response, which tries to barricade these bacteria, fungi, and viruses, failing which then the adaptive immune response are triggered. Of course, as I said, it's seen in only vertebrates. And here you have a humoral immune response and a cell-mediated immune response. Humoral response are the antibodies, which defends against the infection in the body fluids and also triggers the cell-mediated immune response specific to the antibodies. And we have a cell-mediated immune response, which are cytotoxic, kills the cells, promotes the take-up of these antibodies, the invaders, and also helps in killing the infective organisms or the cells which are infected by the viruses or hosts. So We have a 3-level barrier systems, which we call first 2 layers are created by innate defense, and the subsequent layer is created by adaptive immune responses. The adaptive immune response are usually, as we say, are specific, and it needs to be pre-sensitized before they respond, whereas innate immune response doesn't need much of pre-sensitizations. So the major cellular components of the innate non-specific systems, as you know, they have platelets, neutrophils, eosinophils, basophils, monocytes, which evolves into the macrophages. B cells and T cells are the adaptive immune responses, which are evolve into the plasma cells, and T cells into varieties of cells. And as you know, all these cells which place or have their source from the bone marrow subsequently migrated into the thymus, which is responsible for T cells. The B cells are mainly synthesized, produced in the B cells, in the bone marrow. And how the inflammation, how does immune response comes? See, there are 2 terms we use. We use a term called inflammation, and we use a term called immune response. Sometimes they are interchangeable, but most often when we say inflammation, it is a specified response against an altered homeostasis. Earlier, we always used to think it needs a stimuli of the external agents, either the injury, insult, or a bacteria or a fungal infection to happen for inflammation to trigger. But we now know even a minor disturbance in the homeostasis do trigger inflammation, as simple as the psychological distress or even the distress of infections. So those things can also trigger an inflammation. So there are stages or steps in which the inflammation can happen. Inflammation can be very much localized. For example, an abscess or a wound which is only localized to the site of infection, and the whole process will be localized or regionalized into those areas. This is what we call as localized inflammatory response, or it could be systemic. In this, we divide into 2 major steps. One is where you have a systemic inflammatory response like fever. The pyrogen moves out of the local sites, which in turn triggers the fever generating system, which is located both in the brain as well as in the periphery, which in turn increases the overall temperature of the host. That is what we call as a fever response. But that's a systemic inflammatory response may not have a severe aggressive inflammatory response, or sometimes it may go into a life-threatening, overwhelming inflammatory response, what we call septic shock or a cytokine storm, where the cytokines are released in excess to generate a triggered immune response across the host. That is what we call as a sepsis or a septic shock. So an immune response could be localized when it's a perfect way of handling the scenario, may generate a low-grade fever to high-grade fever, may also help and facilitate in the inflammatory or immune response, but may not cause much serious harm to the host, or may go into a sepsis shock where there is a blast, where the entire host is responding to the infections or inflammations, or which in turn may be life-threatening, where the inflammatory response may cause damage to the host. So what's the initial step when it comes to an immune response? As we know, as the pathogen enters, as I said, after it bypasses the layer of innate immune response, the barriers, then the cells, host immune cells, starts recognizing these pathogens. Without a recognition of the pathogen or the foreign material, we don't develop an immune response. The particulate matters which do not trigger or initiate an immune response, we call it as inert foreign bodies or inert to immune response. For example, the steel rods which we insert, some of the meshes which we use for correcting the hernias. These are inert materials. They are not recognized by the immune cells. So since they don't recognize by the immune cells, the host don't mount an immune response on these foreign materials. So as a consequence of which, we can insert these materials to repair the host's fractures, to repair the host's membrane which has gone wrong. So these are called inert materials. Inert materials escapes the initial step of recognition, as a result of which you don't get an inflammation. So, or an immune response. Otherwise, any other structures, any other foreign body, including the living organisms like bacteria, fungus, etc., they need to get recognized. Recognition of by the host of a foreign body, including the living or non-living living particulate materials, is the initial step for developing an immune response. So here we have manipulated to greater extent. We have learned which are inert materials. We are designing inert materials to be used for putting it for a graft, putting it for repair, for putting it for a structural wherever we require. That has us in modifying or altering the initial step, the immune response. So we know how there are varieties of steps in phagocytosis. If a pathogen or a splinter, say, insert, that in turn causes an injury that results in the initiation of a signal molecules, which are released by the ruptured cells, which are released by the splinter or a pathogens, which in turn activate mast cells, macrophages, and through these signal molecules, they start the chemotactic movements, the movements of the fluids, which in turn also permits the egress of the neutrophils and other phagocytic cells, which in turn can make the tissue to gulp them up, digest them. If they are successfully can clear it, there is no role or no room for the second site of immune response. Quite frequently, the innate immune response may handle it in the very much in these points. And if this happens, possibly the body may not get sensitized to these proteins, but most often, if they are a particulate matters or some other materials, if macrophages takes it, they slowly move themselves, these proteins, into the lymph nodes or any other center, follicles, etc., where they try to present these proteins for a future memory with reference to those particulate matters. Again, as I said, it depends on the load of the organism. If the load of organism is very limited or very little, the cells may not have so much of proteins. If that's the case, usually the body do have a mechanism enough not to proceed further in sensitizing the adaptive immune response. That's why if you have said that, you might have seen scientists telling that in an asymptomatic response, in an asymptomatic carrier, sometimes the immune response may not be adequate, may not be long lasting, they may need vaccination. The reason is, if there is no adequate particulate matters, there may not be adequate sensitization of the adaptive immune response. If your adaptive immune response is not sensitized, likely your resistance for a second invasion may not be that great. So now it comes to the adaptive immune response. You know, the cells that are involved are lymphocytes. And there are few terms which I would like to introduce here. We use a term called antigens. Antigens are a little different from pathogens. All pathogens are not antigens. Antigens are the substances that can elicit a response from B or T cells. If they don't elicit a B or T cell response, only elicit an immune response, we use sometimes called immunogens, immune stimulators, etc. But when we talk of antigens, we tell the antigens are the substances which elicit a response in B or T cells. And there are older definitions where we use a term antigen loosely, where we use the term antigen. Antigen is a substance which elicits an immune response. The antigen eliciting an immune response of other than the B and T cells are just immunogens. But when the antigen elicits, the substances which elicits a B and T cell response are the true antigens. And in the lymphocytes, we have two types of lymphocytes. One is a thymus-derived lymphocytes called T cells, bone marrow-derived lymphocyte or a bursa-derived lymphocytes called B cells. Exposure to pathogens activates B and T cells through the antigen receptors. It's not that it recognizes the entire protein, it recognizes only few segments or few part of the amino acids in these proteins, which are otherwise called as epitopes. So these are recognized through what we call as by B cell through B cell receptor, in T cell called T cell receptors. B cell receptors are usually a Y-shaped molecule resembling the immunoglobulins. They are like two identical heavy chains and two identical light chains, whereas T cell receptor have an alpha chain and a beta chain, which are not identical. They have something what are called variable regions, which alter in its configuration, making it to be a lock and key mechanism with the antigens. So the variation in the antigen binding sites make them unique, and these are what are called as repertoires, which in turn matches to the epitope, the amino acid sequence which are present in the antigens. So the constant region of the chains are very little. They don't vary among the B cells, but they vary between the varieties of immunoglobulins, what we call G, M, A, and E. And the variable regions are the one which alters to suit to the epitopes. So how do they differ, the B cells and T cells? B cells binding B cell antigen receptors is an early step in B cell activation. This in turn gives rise to cells that secrete antibodies or immunoglobulins. They mature into plasma cells, and once they mature into plasma cells, they secrete B cells. Whereas T cells, these are identify antigen fragments bound to the cell surface protein called as MHC, and through MHC only the T cell identifies the antigen. If antigen is free lying outside, the T cell receptor will fail to identify antigens. Antigen always and need to be always loaded on major histocompatibility complex, that's otherwise called HLA molecules. Once they are loaded, then only they are going to, the T cell is going to identify. So T cells have a restrictions. It cannot identify antigens which are freely lying outside. Whereas B cell receptor can sometimes bind to the free lying antigens, and which in turn are taken up by the antibodies. It gulps them in and subsequently can present it as an antigen presentation. So B cell has a unique property of even sometimes identifying free lying antigens, which of course it prefers it to be presented through MHC, but it also can identify a free lying antigens, which in turn has a double advantage of having a quicker response to the antigens much before it is taken up by the antigen presenting cell and presenting to T cells. So you look at the way the nature has evolved, because sometimes if the infection is too aggressive and too fast, rapidly spreading, waiting for a T cell to get identified, recognized, and then going through the process of identification, etc., is a time-bound process. To cut short, there are shortcuts which has been identified with the B cell works. And there are a lot of differences these B cells and T cells have with reference to their epitope spreading, hypervariability maturation, hypervariability mutations. They have made these two cells to be unique, and that B cells and T cells interact each other to have a regulatory mechanism so that they don't overreact at the same time, they don't have an under response. So based on these principles, we now have recognized that there is something what is called as the immune responses goes what is called as learning, training, and adaptations. And both we till last a decade or a decade plus, we were thinking that innate immune response is a very non-specific response, and they don't have anything what is called as learning or a memory. And it is that they are ready as soon as they come, they fire and trigger it. But we have learned that even innate immune response goes for an adaptive changes. It changes, it keeps its trigger either low or it keeps its trigger high based on the host's exposure to the viruses and proteins. It also gets based on the host's psychological stress and social stress. And this has been very clearly shown in human beings who exposed to psychological stress, who exposed to physical stress, they alter, they get either activated or they get suppressed down, or we call them something what is called as trained memory and they are not antigen specific. Vaccines do sometimes go for something what is called as trained memory or keeps the innate immune response little high. So this is one of the things which the vaccinologists, when we give a vaccine, even if it doesn't have a specific spike proteins of the next new strains, may help and may benefit in giving a sort of a protection against the viral infections. So it may not let the virus infection to be very aggressive because innate immune response is in an already heightened active stage. What we meant is, though we really don't know who is the likely enemy, though it can identify through the pattern recognition molecules, the response may not be unique to that virus, may give a non-specific immune response, a sort of a trigger happy firing. So you don't know who the virus is or who the person who is moving around, but you have a gunshot to shoot, so you shoot it. You can shoot it. So if it is right and if it is lucky, the things works for the host. So that's how the innate immune response goes to a trained memory or a sort of a non-antigen specific rise or in its activity, and also have a sort of a not antigen specific memories wherein they altered the expression of the pattern recognition molecules. They may hyperexpress TLR5 based on what are the likely antigens. They may hyperexpress TLR9 based on the expected antigens based on the host perception of the external environment. Whereas adaptive immune response is something we have a diversity in the lymphocytes based on the receptors. So they may have a lack of reactivity against the self-owned molecules or may have a trigger a specific immune response. Only when the lock and key gets suited, the B and T cells proliferates and gets activated. And here you need an immunological memory. It is antigen specific. It is antigen specific to certain point. Afterwards, the antigen specificity loses. What we meant is once the T cell gets activated, the secreted cytokines, secreted action. For example, once the locking occurs, if the interferons are produced in excess, the interferon action on the subsequent cells are not antigen specific. It is what interferon can do. So though the initial action is antigen specific, the subsequent reactions are not necessarily antigen specific. These are the immune pathways which otherwise when T cell gets activated or triggered happens. So that's how, so we need to know that in an immune response, some steps are specific, some steps are non-specific, some steps which are antigen specific can be manipulated based on the antigen manipulation. Some specific steps which are not immune response specific can be altered by a non-specific inhibition and non-specific immune response. For example, you might have heard that tofacitinib anti-IL-6 therapies were useful in preventing cytokine storm. These are all non-specific inhibitors. They blocks the IL-6 response, which would have non-specifically triggered a mounting cytokine response. That itself could be a way how one can manipulate or alter the immune responses. So when an immune response on an infection or for that matter on any disease, we consider it as a failed immune response when the immune response fails to defend against either microbes or some cancer. Say, for example, it allows cancer cell to proliferate. It has failed to inhibit the cancer cell. If it has failed to inhibit the microbes or failed to kill the microbes, so it has failed to or adequate mount the immune response. Immune response also can fail when there is a hypersensitive reactions, when it is out of the control response like a cytokine storm or a sepsis, or when there is a immunodeficiency, you may also land up in the failed immune responses. So now if you look at how a minor things, this is just a representative of one or two concern, but it is not just one cell, multiple cells could be responsible for this. For example, now let us just take Treg cells. Say if there is not adequate Treg cells, if the Treg cells are less or there is natural Treg cells are less, and there is a blockade of Treg cells through IL-10, TGF-beta or CTLA-4. There is an enhanced T cell functions. So when you have a enhanced T cell regular functions, the microbes induced pathology increases. The microbes can proliferate, and the pathogen can expand. And if there is a persistent infections and the immune response is good, adequate, because the Treg cells are not influencing negatively on the infection elimination, slowly pathogen expansion is brought down, and there could be infection persist for some time. Slowly the pathogen elimination occurs. Once the pathogen elimination occurs, you don't require any more immune responses. So immune response should slowly die down. This is where the Treg cells promotes. Treg cells increases in number. They go for an enhanced Treg cell functioning, and the immune response should slowly come down. So this is the best immune response, what I can anticipate. On the contrast, suppose Treg cell function is in excess here, and you don't get an adequate immune response. What really happens is microbes expands, pathogen expands, infection persists. And once the infection persists, what really going to happen is the infection spreads across the host. On contrast, if you don't get an adequate Treg cell functions, the pathogen triggered immune pathology persists. The cells are going to secrete cytokine excess, cells are going to be cytotoxic, which is going to cause even the host damages. As I said, the pathogen might have been eliminated by antibacterial or any other interventions that we have done, but the immune response that has been triggered may be rolling down. So the immune response that is rolling down is going to cause the damage to the host if you don't get an adequate Treg cell functioning, wherein it should downplay the immune response that has been triggered. Just to give a simple example, you should be angry, you should put all your adrenaline to fight an enemy. After the enemy has gone, you should reduce your adrenaline surge and put down your arms and ammunitions, quiet down. Otherwise, if you continue to swing the arms, shoot the guns, what you are going to damage is your own people. Because once you have killed the enemy, there are no more enemies in that place. So if you continue to shoot, who gets hurt are your own people, or the people who otherwise need not be killed or damaged. So that's what happens. If T regulations don't come, the immune pathological process that has been triggered by the pathogens continues to onslaught. That could result in the what we expect as a damage to the host by the immune pathogens that has been triggered. So finally, as I said, the host response depends on the immune status of the host, type of organism and its virulence, and other factor. The final response, several host factor influences, for example, the environment, which could keep your immunity up or down, age, gender, because as you go elderly, the immune responses are shunts down. In the younger age group, immune response is yet to mature. You see them in the best in the middle age, and their lifestyle, physical activity, smoke, etc., vaccination status, other infection status, co-infection like HIV, CMV, EBV, and other viral infections, co-infection status, they all keep the immune status in a different state. That itself can influence the way the body gives an immune response if the immune response is not perfectly balanced. For example, activation, adaptive immune response, post-threshold, we expect the expansion to go for a contraction and inactivation. If anywhere these things fails, the situation may result in the development of progression of immunopathogenesis, even triggered by infection, autoimmunity or self-immunity when these threshold crosses may continue to cause the damage. So that, to put it in a nutshell, the need of our is to better understand the immunoregulation and pathophysiology of both autoimmunity, infections, as well as what happens in post-MI, post-stroke. These all are going to help us how to reduce the damage that is triggered by the initial event. Subsequently, you have both, you need them, you can't stop them, you need that action, but after the action, we need to know when to stop it. Sometimes you need to fire a gun. When you have a terrorist standing right in front of you, saying that don't fire the gun is going to be a misadventure. I think we have done that in our history. And when there is, after the things are done, you should know when to stop the fire. That's also crucial for things to work out. So it's a need or a necessity for us to trigger a war, and we need to know when to stop the war. So this is what we need to understand in immune system is, it's one of the very well-balanced, highly regulated, well-organized system. But because of the complexity and because the way it works sometimes, the way it works on a dynamic manner, it's an orchestrated effort. It's not that one-man army. It's multiple players play with it. I always keep telling my students and the people who want to understand, it's like a coalition government. It needs a perfect balance. Anywhere pulling here and there can cause a lot of trouble. So we need to understand these steps. And once we understand these steps, since we now know how to intervene, probably intervening in the right time is going to help. So adding to that, we have a lot of confusing terms. We use term called immune boosters. We use term called immune regulators. We use immune suppressants. And of course, we have a vaccines. Vaccines, we have two variants, which boosts it, which alters the immune responses. We have something called desensitization, which we use in allergy and autoimmunities. And we have several of these terminologies, which we really have to understand. Are we really doing something called a boosting? Are we really regulating the immune system? Are we really suppressing the immune system? What really makes things different? So we need to understand these perspectives for us to work towards in getting a proper regulations of the immune system. And if we know how to regulate the immune system, how to work towards it, that will help us in manipulating most of the diseases where we lose the patients. We have chosen a right antibiotic, patient is doing very well. We see suddenly patients start deteriorating because of the immune response has not been blunted. So also it happens in autoimmune disease when we use immunomodulators or immunosuppressants. After a certain point, when the patient starts getting infections, we really need to know where to block it. So it's still a system which is to be evolved, and we have a lot to understand. We can say at least we have understood to a greater extent in the last 2 decades, in the last 20 years, but there is much long way to go. Hopefully in the years to come, I could see a lot of youngsters sitting across who can put their mind in learning, understanding these processes, and getting the correct guidelines, how, where, and how to intervene so that we have a smooth flow. When you wage a war, you should know when to command yes, when to command no, but to know that you should know the complete war game. Thank you very much for patience hearing. Any questions? I think in next 10-5 minutes.
Participants, if you have any questions, please unmute yourself and ask the question. Or you can drop it in the chat box. If there are no questions, I would like to thank Chandrasekhar sir for starting the talk with basics of immunology, that is, what are the invaders of the system, followed by the types of immune system, innate and adaptive system, and the responses for the invaders. Thank you, sir. Thank you so much for your talk.
Thank you.
Thank you, sir. I request all the quiz participants to come to the auditorium for the quiz competition, and I'll post the link for the feedback form please fill the feedback form. I request all the participants to fill the feedback form. The link is provided in the chat box. I request all the audience to fill the feedback form. The link for the feedback form is posted in the chat box. And I request all the quiz participants who are online, please assemble in the auditorium for the prelims. I would like to bring to the kind notice of the participants, you will get your feedback, you will get your certificate only if the feedback form is filled. So please fill the feedback form since the feedback form and your certificate is connected with each other.
You cannot break your. I can. I have done many times.
I request all the audience participants of the quiz to please assemble in the auditorium. I would like to mention again, the link for the feedback form is provided in the chat box. Please fill the feedback form. With this, we come to the conclusion of the technical session. Thank you one and all. Thank you. See you.